Welcome. Welcome, my dear friends. Myself, Professor Dr. Rajendra Deshpande, would like to welcome you all in this interesting video regarding the physiology subject. Physiology, and we are discussing today about the muscle physiology. This lecture will be definitely useful for BAMS, BHMS, homeopathic, physiotherapy, and also MBBS students. So today we are going to discuss about molecular basis of muscular contraction. Okay, so myself is Professor Dr. Deshpande. I am MD in Ayurvedic medicine and MD in Ayurvedic physiology, that is Kriya Sharira and Kaya Chikitsa. So let us start to discuss about the muscle physiology and especially muscle contraction. When this muscle contraction takes place, what are the actual molecular level activities takes place that we are going to discuss? Okay, so this particular action is responsible for the actomyosin complex. Try to understand, actin and myosin, these are the filaments in the muscle, and these protein filaments are responsible for muscle contraction. So together, actin and myosin, that particular actomyosin complex results in the muscular contraction. But of course, it is a step-by-step procedure. So first stage is excitation, contraction, coupling. There will be stimulus, excitation, then the contraction, and together is called as a coupling. In this particular process, what is the role of troponin and tropomyosin? These are again protein molecules in the actin filament. Okay, and then we will see how that particular actin and myosin are overlapping each other. Myosin is stretching to the actin, and this particular process is called as the sliding mechanism. Okay, so first, excitation, contraction, coupling. This is the process occurs when actually the excitation, suppose for example there is some stimulus from the brain, neuromuscular junction, the signal is going, motor activity is starting from the brain and it is giving signal to the muscle. What signal? About the contraction of the muscle. So first there will be excitation and then there will be contraction. This particular process, although I am saying in one sentence, there is a series of activities. There are different activities which are responsible for contraction of excited muscle. Okay, when a muscle is stimulated, or excited, that impulse coming from the brain come through the motor nerve to the neuromuscular junction. Some words are very important. Neuromuscular junction, connection of the nerve and the muscle, Then there will be action potential is generated. First, the muscles or any cell is always in resting membrane condition. All time they are resting. But when there is something stimulus, they start the action, and that particular energy is called as a action potential in physiology. This action potential spreads over whole process of sarcomere in the muscle and also into the muscle fiber through the T-tubules. This is the particular structure. I am going to explain it afterwards. What is the T-tubule? The T-tubules, that is the transverse tubules. T means transverse. T means transverse. These are responsible for rapid spread of that particular current of action potential into the muscle fiber. So what is the role of T-tubules? It will carry the action potential to the muscle fiber. First starts in the brain, then neuromuscular junction, and then to the muscles. Okay, next. Now first try to understand the basic terms in the muscle, because you know the sarcoplasm. This is the structural and functional unit of the muscle where actually the actions takes place. So what are the T-tubules and L-tubules? I am going to prepare another video also for that matter to explain you the structure of muscle. But T-tubules are nothing but a transverse tubule, narrow tubules formed by the invagination of sarcomere. Okay, these tubules penetrate all the way on one side of the muscle fiber to the another side because it is transverse. T-tubules are responsible for, as I have said, rapid transmission of impulse in the form of action potential from sarcomere to myofibrils. Okay, this much you have understood. Now, what are the L tubules? L is a longitudinal. It is also called as a sarcoplasmic reticulum. L means sarcoplasmic reticulums. These are the closed tubules. T tubules are open. These are the now closed tubules run along the long axis of the muscle fiber, forming sarcoplasmic reticulum. And what is the function? L tubules. Here you can see the large quantity of calcium. Calcium is very essential ion for muscle contraction. Don't forget calcium. That's why we always stress to the people, youngsters, that take some milk. Milk is a high source of the calcium. So calcium deficiency always cause the problem in the muscle contraction. There will be twitching, there will be tetany. So to get the proper action of muscle contraction, the person will need calcium. And that is stored in this particular L tubules. Next. Now, what are the cisternae? This is a typical term in again that particular structure of the muscle. At regular intervals throughout the length of the myofibril, the L tubules, longitudinal tubules, dilate to form a pair of lateral sacs here and here, pair of lateral sacs which are called as terminal cisternae. Each pair of terminal cisternae is in a close contact with the tubule. The T-tubule, transverse tubules, along with the cisternae on either side, are called as triad. This is very important short note for the students. Actually, the LAQ is muscle contraction. Describe muscle contraction. Describe the sliding filament theory. These are the long questions for 10 or 15 marks. But there are some short notes, SAQ of 5 marks or 3 marks, that is regarding the triad of skeletal muscle. Now, calcium ions are stored in the L tubule, longitudinal tubule, and the amount of calcium ions are more in the cisternae. Next, when the action potential reach to this particular cisternae of L tubules, these cisternaes are excited. Now, the calcium ions stored in the cisternae are released into the sarcoplasm. Okay. The calcium ions from the sarcoplasm move now towards the actin filament to produce the contraction. So first there was action potential, then there is a calcium release, and now the process of action potential. Excitation of muscle fibers by impulse passing through the motor nerve and neuromuscular junction, generation of action potential in muscle fiber, spread of action potential by T-tubules, entrance of action potential in cisternae, release of calcium ions from the cisternae, movement of calcium ion towards the actin filament, and then the contraction of muscle fiber. This is like in short summary what you have understood till now. Okay. This structure you can see, these are the myofibrils. Just I am showing you, these are the transverse tubules. Can you see? Transverse tubules. Then this is sarcoplasmic reticulum. Then here you can see the cistern. This compartment is called as a cisternae, dilated part. Okay. And along with this both side, cisternae and T-tubule, they are called as a triad. Okay. Next. Here you can see this is the sarcolemma. Then these are the mitochondria. These are the myofibrils. Then here you can see again the T-tubules, transverse tubules, terminal cisternae, and the triad. Sarcoplasmic reticulum. This is the structural and functional unit of the muscle. Now, the calcium ions forms the link or coupling material between the excitation and the contraction of the muscle. Okay. Hence, the calcium ions are said to form the basis of excitation, contraction, coupling. This can be the MCQ. There will be MCQ. Dash ions are said to form the basis of excitation and contraction on the calcium ion. There will be difference like sodium, iron, potassium. Don't get confused. Calcium ion is important for muscle contraction. Now, the role of troponin and tropomyosin. Normally, the head of the myosin molecule has a strong tendency to get attached with the active site of F-actin. Okay. However, in relaxed condition, the active site of F-actin is not open. It is covered by the tropomyosin. This is the always they will not allow the myosin this is a big myosin, is a big filament, strong filament to come to the actin. They will not allow. Unless and until there is necessity of contraction, actin molecules are little bit shy and they are not open. Okay. Therefore, the myosin head cannot combine with actin molecule. Okay. Now, large number of calcium ions which are released from the L tubules during the excitation of the muscle bind with the troponin C. C is named for the calcium, where the you can find that calcium entry can occur. The loading of troponin C with calcium ions produce some change in the position of troponin molecule. Now there is a configuration will be changed. Configuration will be changed in actin molecule. Now here you can see tropomyosin molecule is there. This is troponin. Okay. This particular configuration, first it is closed, but when there is a calcium ion release is there, then myosin are welcomed by this actin molecule also. Okay. So this particular figure you have to draw of actin filament. Now next, role of troponin and tropomyosin. It is in turn tropomyosin molecule away from the F-actin. This is taken away. Due to the movement of tropomyosin, the active site of F-actin is uncovered and exposed. This step is very important. What is important? In the actin molecule, configuration is changed. In actin molecule, configuration is changed. Why it is changed? Because head of the myosin should come in contact with actin. Are you getting why there is a change in configuration of actin? Head of the myosin should come near to the actin. Immediately, the head of myosin get attached to the actin. Now, sliding mechanism and formation of actomyosin complex. This is called as a sliding theory. Sliding theory explains how the actin filaments slide over the myosin filaments and form the actomyosin complex during the muscle contraction. This particular theory is also called as a ratchet theory or walk along theory. Walk along, sliding or walk along theory. Okay. Each cross bridge from the myosin filaments has got three components. That is, we are now discussing about the myosin. Hinge is there, arm is there, and head is there. Okay. Here you can see this is the head of the myosin, and this is a actin binding site. Okay. This is for ATP. A diagram showing the myosin filament. ATP is nothing but the adenosine triphosphate. This is energy molecule. After binding with the active site of F-actin, the myosin head is tilted towards the arm so that the actin filament is dragged along with it. Myosin head is dragging to the actin filament. Now, you can see over here, this is the myosin head. These are the actin filaments. Okay. This is the ATP and this is the hinge. Here you can see the hinge. Okay. And this is myosin. This is myosin. This is actin. This is the head of the myosin. It is attached to the actin. And now it is pulling. It is pulling toward near and near. Both sides of the myofibrils are coming nearer for the contraction. Can you see in the contraction there is coming near is very important. Okay. This tilting of the head is called as a power stroke. I will go back again. I will show. This is called as a power stroke. This is power stroke. What is that doing? Head of the myosin is pulling to the actin. Head of the myosin is pulling. This is called as a power stroke. Okay. After tilting, the head immediately breaks away from the active site and returns to the original position. Now it combines with a new active site on the actin molecule. So one by one, again I will show you. After this, it will be released. Again, it will go here. Again, it will be pulling. Again, release. Again, pulling. Again, releasing. This is a continuous, what you can say, the cycle. Now it combines with a new active site on the actin molecule and tilting movements occurs again. Thus, the head of cross bridge bends back and forth, and pulls the actin filament towards the center of sarcomere. Where it is pulling? Center of sarcomere. In this way, all the actin filaments of both the ends of the sarcomeres are pooled. So the actin filaments of opposite sides overlap and form the actomyosin complex. Formation of actomyosin complex results in contraction of the muscles. Here you can see the structure of myofibril. Each myofibril is consists of alternate dark A, can you see here, dark A band and light I bands. In the middle of the A band, there is a light area called as H zone. Can you see H zone? Again, this is pink in color. What is H? H is a German word. The meaning of H is hell. Is not English. Hell means light. This is the German language. And H is also given the name because of the discoverer, that is Henson was the discoverer who find out this particular research. Okay. In the middle of the H zone, in the middle of the lies the middle part of myosin filament, which is called as M. Here you can see M line. This is the central part of the H zone. In the German, Mittel means middle. Mittel means middle. M line is formed by myosin binding proteins. Okay. Sliding mechanism and formation of actomyosin complex. This is sliding theory. When the muscle fibers shortens further, the actin filaments from opposite side of the sarcomere approach each other. So the H zone becomes narrow. Again, I will go back and show you H zone. Can you see the H zone? This H zone becomes smaller. H zone becomes narrow. And the Z lines come closer with the reduction of the length of sarcomere. Can you see again? I will go back. Z lines. Concentrate. These Z lines come together. Z lines come together. Okay. Then, however, the length of A band is not altered. That is very important. That is the question for MCQ. However, the length of I band also decreases. Now, here you can see the structure of muscle. Z lines come closer in contraction. This is the contracted muscle and this is the relaxed muscle. Can you see this is Z line, this is Z line, they come closer and this is away from each other. No. Sliding mechanism and formation of actomyosin complex, sliding theory. When the muscular contraction becomes severe, the actin filaments form, opposite sides overlap and the H zone disappears. Changes in the sarcomere during muscular contraction. These changes that takes place in the sarcomere, structural and functional unit of the muscle during the muscular contraction are: number one, length of all the sarcomeres decrease because the Z lines come close to each other. This is for MCQ. Which line comes together? Length of the I band decreases since the actin filaments from opposite side overlap. H zone either decrease or disappears. And length of the A bands remains the same. This is the question for MCQ. Which band remains the same? A band remains the same. Okay. Now we will go further. Sliding mechanism. Release of large amount of calcium ions from cisternae of L tubules. Binding of calcium ions with troponin. Change in the position of troponin. Pulling of tropomyosin molecule away from the F-actin. Exposure of active sites of F-actin. Attachment of myosin head to the actin. Power stroke, as I have showed you, power stroke. Tilting of the myosin head and the dragging of actin filament. Detachment of myosin head and returning to the original position. Detachment of myosin head and returning to the original position. Next, what is the next step? Attachment of myosin head to the new active site of F-actin and dragging of actin filament. Repetition of these actions. Movements of actin filaments of both sides towards each other and formation of actomyosin complex. And finally, this is the contraction of muscles. Of course, you know, after the contraction, there must be relaxation. So relaxation of the muscle occurs when the calcium ions are pumped back into the L tubules. Calcium is taken back because the necessary action has been taken. When the calcium ions enter the L tubules, calcium content in the sarcoplasm decreases, and that leads to the release of calcium ions from troponin. Okay. It causes the detachment of myosin from actin followed by the relaxation of the muscle. The detachment of myosin from actin obtains energy from breakdown of ATP. So, the chemical process of muscular relaxation is an active process, but physical process is said to be passive. This is also for MCQ. because there is energy, things are happening. So chemically, it is an active process of muscle relaxation, but physically, it is the passive process. This can be the MCQ question. Okay. Along with the other proteins and some enzymes, actin and myosin form the molecular motors which are involved in the movements. So pumping of calcium ions into the L-tubules, decrease in the sarcoplasmic calcium content, release of calcium ions from troponin C, detachment of myosin head from F-actin, and muscular relaxation. So my dear friends, I hope that you have liked this video of explaining the molecular basis of muscular contraction, sliding filament theory. This is a very important question for your examination. So please, I'm putting different videos on the physiology also on my YouTube channel for the BAMS, BHMS, homeopathy, and physiotherapy students. Please like this video, share this video, and don't forget to subscribe my YouTube channel and press the bell button icon. Thank you very much, and all the best for your exam and for your career. Thank you very much.